Hey guys, welcome to our third session of Living Environment Regents Review. Thank you guys for being here today. Those who are following us on stream, thank you for keeping up with us. Over the three sessions, our first session, we took a journey through the cell where we went through all the different cell organelles. We explained the life processes that's taking place and how they interact together. We went through the cell membrane. We covered some biochemistry. We went into the mitochondria to review cellular respiration. We covered the ribosome as far as protein synthesis. We covered chloroplast, the process of photosynthesis. And we also explained the role of the nucleus in the process. To end that first session, we spoke about the centrioles and their role that they play in reproduction, in cell division for mitosis and meiosis. In our second session, we spoke about how the reproductive process takes place. We reviewed the male and female reproductive system, which took us through development. From development, we spoke about how traits were inherited. And so now in our last session, now that we've gone through all the body systems, we're going to wrap it up by talking about how you inherit the traits that you've inherited in the process of development, and how the abiotic living things and non-living things interact in the environment. So once again,
It's on the second slide.
Okay, so that's gonna take us into our session. So here we go. So for today's session, we're going to look at sexual reproduction, quick review, go over plant anatomy, how that's related to our anatomy. Some of the processes are the same. We will cover basic Mendelian genetics, just a few of the laws that I've seen over the years on the Regents exam. We'll cover gene variation. We'll touch on a few of the genetic disorders. Definitely a big topic on the Regents is biotechnology, so we'll spend some time talking about that. Under biotechnology, you'll see GMOs, cloning, DNA fingerprinting are very big topics that we usually see questions on. Darwin's theory of evolution, dealing with natural selection, we'll cover that. From there, we'll go right into human impact on the environment, how that affects biodiversity, and how the environment is changing over time through successions. Okay, so let's get into it today. So here's your quick review of fertilization. We have the fusion of the egg. Here's the male gamete, is another name for the sperm cell and the egg cell. Once the nucleus fuse together in fertilization, you create the zygote. The zygote is where you have both genetic information combined so that the offspring has everything it needs to survive. So it's kind of the union of fusing that genetic information and ensuring that the cell has all the chromosome, all the genetic information that it will need. Once the cell is fertilized, here we go, A represents the zygote. The cell will go through a series of cell division referred to as cleavage. And this cell division process is controlled by mitosis. Once that process takes place, we will notice that the cell will divide into many different cell layers, such as the ectoderm, mesoderm, and endoderm. Each layer, based on their location, will be expressed into different genes or different tissues, different cells, to form different tissues, which eventually form into the different body systems. Okay. So we went on to differentiation. This is where we left off the last time. Here's the zygote. Differentiation is that process of creating the skin cell, nerve cell, and muscle cell. So this is the process that transforms developing cells into specialized cells with different structures and function. Cells maintain identical genetic information. So every cell, going from the zygote to all the different types of cell, have the same genetic information in it. The expression of the skin cell and nerve cell, all the different cells in our body is derived based on their location. Depending on their location, the gene will express in different ways based on their location. I could be in? Okay.
Can see you.
Oh, I was trying to stay out. All right, so here we go. We have the ectoderm, which we said would be the outer layer, where this will give rise to your skin cell, nerve cells, pigment are a few of the many cells that you will see. The mesoderm, on the other hand, that middle layer, will give rise to the red blood cell, skeletal muscle, and smooth muscle, to name a few. Whereas the endoderm, is that inner layer which gives you the lung, thyroid cell, the pancreatic cells, are just a few of the cells that will derive. The main thing you guys need to focus on for the test that we're preparing for is the fact that cells derive or differentiate that into different cells. The zygote will differentiate into different body cells through differentiation. So even though you have that same genetic information, due to gene expression, you will see different cells develop. All right, so expression of an allele or a gene can be influenced by other genes themselves or other things. So we just saw in the development of that zygote that the location of the cell made different cells, created different cells. Like if you were outside of that gastrula phase, those cells became the skin cell of the body. If you were on the inner layer of that developing cell, you would become some of the organs and such. But there are other things that can influence gene expressions, not just in the zygote, but we could see temperature playing a role. We could see environment playing a role. This is where we get the idea of where we have identical twins. Imagine that they would grow up in completely different environment, exposed to develop into different environment. We could see that those twins can express differently or arise with, not that they have different traits, but the expression of the genes can be expressed differently because of the location in their environment and what they're exposed to. We see that in plants. Plants, during the fall, we see different variation of colors in the plant. That's triggered by the change in temperature. So sunlight can affect the expression of genes, location, nutrients. These are all factors that can affect it. Here are a few examples you may have covered in class. The Himalayan rabbit, that was the example where they shaved off the fur off the rabbit and exposed it to lower temperature, and we saw that due to the lower temperature, it triggered that gene to produce black fur in the animal, because black fur would absorb more heat, keeping the rabbit warm. So in lower temperature, that rabbit has dark fur. See it in plants, as I express, that light will trigger different types of chlorophyll. Twins in different environments, and we talk about those stem cells. We just saw in the zygote that they code for different things based on where they're located. Right? So let's take a quick look. Here's a quick practice question. So this is page 1 of our review sheet. So take a second. Look at it. The data table below summarize the result of an investigation in which seeds from the same plants were grown under different conditions and temperature at relative humidity. So if we take a look, we have plants that are exposed at 20 degrees Celsius, humidity at 20%, plants at 31 degrees Celsius, humidity at 95%. Here are the genes, and this is how they appeared in the organism. Here are the same genes again, and this is how they appeared in that organism. So which conclusion can be drawn from the information in that data table? I'll give you a second to think of that. What would you guess based on the result? See, what is causing the change in the genes? So from here you would see that it's the environment. So a conclusion that can be drawn here is that the environment influenced the expression of the genes. Whereas here at low temperature, we see that different colors appeared in the organisms. Whereas, at higher power, So in a higher power, higher temperature and humidity, those genes are expressed with different colors. So the environment will influence it. So once again, we're reviewing from last session and to lead into our next session. So here we see that each one of those cells that arrive through differentiation, whether it's skin, muscle, nerve cell, all of those will then work together to form different tissues in your body. So those muscle cells, that's where we get skeletal muscle tissue, smooth muscle, cardiac muscle tissue, and that tissue will then form an organ. So the cardiac tissue would form the heart, which is an organ in your body. The organs will then form a system. The heart is a part of your circulatory system, which we saw. So to show how they're connected, we could see that through digestion, we have the nutrients, all of those body systems, the nutrients digested in the digestive system, diffuse into the circulatory system, where it's then carried throughout the body. So we're showing now how they work together to maintain balance. The excretory system, in that system we have the kidneys, and the kidneys is where that blood will then get filtered out. So we'll have waste products filtered out, we'll have urea, some water, some salt, and that's where urine is made. From the kidneys, the waste product in the form of urine will be stored in the bladder and eventually gets released from the body. We discussed the respiratory system in the past. The respiratory system is where gas exchange takes place. So this is where that oxygen that you breathe in will then diffuse from the alveoli, the air sacs from the lungs, diffuse into the blood. The blood will take it around the body to where respiration is needed. So that oxygen that we breathe in will end up going to the mitochondria to carry out cellular respiration to provide our body with energy. The endocrine system, we've been talking about different glands secreting different hormones. The hormones will leave from the glands and go into the blood, where the blood will transport it to a target cell. We remember LH, FH go into the ovaries, and triggering the ovaries to secrete estrogen and progesterone to the uterus in preparation for the implantation of the zygote. Then we saw in that state lab of making connections, how does the muscular system, what's the role of the muscular system in the homeostasis or the balance of all of these systems working together. And we see that gas from the respiratory system, nutrients from the digestive system will go to your muscle cells and provide them with energy so that they could perform action, muscular activity. The immune system we quickly glanced over. The immune system, the key things to remember from the immune system are those white blood cells. The white blood cells trigger an immune response, and by doing that we see the T cells came into play in the B cells. So the T cells will attack and destroy. The B cells, they will help in the production of antibodies. The memory B cells will pretty much remember how to produce those antibodies just in case you're exposed to that pathogen again. All right, so let's take a second and do a review of this by choosing a system, two systems, and let's explain how do they interact together. So take a second. We have this at the bottom of page 1. Try to answer this on your own. Those who are following us by stream, I want you to just take a minute. We'll leave this up. Could use a Word document or on a piece of paper. Take a minute to answer this question. Select two systems, identify each system, and state one function of that system. Then explain how the systems together work together to maintain homeostasis in the individual. Let me give you 2 minutes on that. Okay, so let's take a look. If you chose set 1, pair 1, we have the muscular system and the nervous system. So the way they're connected, we have nerve impulses will be sent to the muscular system, send a stimulus to the muscular system to cause movement. And we see that in the reflex arc. You reflex, if you touch something hot, you move right away. That impulse is sent out to the muscle to contract and move. Number 2, we have the respiratory system and the digestive system. The way they're connected, the respiratory system will diffuse oxygen into the blood. We have the digestive system that will send nutrients into the blood. And together, the oxygen will help to release the energy from the food through a process called cellular respiration. Third set, we have the circulatory system and we have the excretory system. So we could see they're connected as far as the blood will go to the excretory system, specifically to the kidney, where it will get filtered. All the waste materials will get filtered out. So take a second, double check your answers, make any changes if you need to. So in the plant, we could see some similar life processes or, processes taking place. In plants, transport takes place by way of the xylem and the phloem. The xylem transports water throughout the plant, while the phloem transports nutrients throughout the plant. So these are vessels that you would find in the vein of the plant that runs through the leaf and down through the stem itself, all the way down into the root. We have gas exchange that takes place in the leaf itself. The stoma, the stomates, there are small openings in the bottom of the leaves. On each side of the stoma are guard cells, and they regulate the opening and closing of the leaves. So a plant that would be in a dry area would not have a lot of stomates because you don't want the plant to lose a lot of water in a dry area. Whereas in an area where, like a rainforest, somewhere where there's a lot of humidity, yet a lot of rainfall, you'll find plants with more stomates to cause that transpiration process to continue. So the guard cells, once again, they regulate the open and closing of the stomates to get gases to go in and out. So that's like our respiratory system, but in plants they have the stomate and the guard cells. Reproduction in plants, we see plants have the stigma and the anther and filament, and over here we have the oval. And so pollen will be found on the anther here, and you could have self-pollination in a plant where the pollen will go here to the style and pollinate the ovals. So eventually in a plant, this would become the fruit of the plant, and the leaf will eventually fall off. So plants do carry out a lot of the processes that we see in an animal. Nutrients are carried out through photosynthesis, which we've covered. That takes place in the leaf of the plant, where we see chloroplast, and it will carry out the photosynthesis in the chloroplast. All right, and that brings us to how are these traits inherited? So we see how the embryo goes from one cell to having to be a multicellular organism made up of all these specialized systems which make up the entire organism. How do you inherit the traits that you have? So traits are the characteristics of the organisms, which is determined by genes. And remember, your genes are coded by your DNA, which is found in the nucleus. And so if you look at the chromosome itself, along the chromosome, you'll have different genes. So these are common images to see on the test, where here's the chromosome and the lines are representing the different genes that's found on your chromosome. I just want to quickly go over a few basic laws. Rarely do I see questions discussing Mendelian genetics on the test that you're preparing for, but you do have to understand the basics in order to discuss some of those inheritance questions. So one of the laws of inheritance is the law of segregation. So you already understand this because we've covered meiosis, but we want to reiterate it again here, what's exactly happening. So the law of segregation states that alleles or genes are separated from each other during meiosis and then recombined during fertilization. So we see that, remember there was that, think about meiosis, you have that cell, then the cell is going to go through cell division. Remember anaphase, prophase, metaphase, anaphase, telophase, goes through it a second time. Then at the end you're going to end up with 4 cells, which is the egg or the sperm if it's in a guy. And each one of those cells will have a set of chromosomes. So when those chromosomes, so here's the egg, pretend this was oogenesis, here's spermatogenesis. During recombination or fertilization, those traits will randomly combine at any point. And so we have here meiosis, and then this is showing how those traits would combine in fertilization. So the characteristics of an embryo is determined by that combination. So what traits you'll get is randomly distributed in the ovary and the testes. You don't know what genes are going to end up in what sperm cell or what egg cell. And so that brings us to the law of independent assortment, meaning that genes for different traits are separated and distributed to the gametes independently. So that first one is saying that the traits will separate, but the independent assortment is saying that they will be sorted independently, not known. So if you take a look here, we have the F1 generation. Here we have the parent cells. We're looking at P cells in plants. Say we have one that's round in color. So we have, it's round, dominant traits for round, dominant traits for yellow. Here, green and wrinkle seed. You have the recessive trait, which is green, recessive trait for wrinkle. And so take a look at their offspring. They could have produced seeds that are round and yellow. But if we take a look in their genotype, we see that they're carrying the traits for round from this parent, randomly carrying the traits for wrinkle from here, yellow from this parent, and carrying the traits for wrinkled from this parent. So it's independently sorted. So it's like taking this deck of cards, here's another deck of cards, and sorting them together. And this is one possibility, a combination from the two. And here, if this seed now was crossed with another seed of the same kind, once again you could see that it would be separated randomly. So where we could have the big R could pair up with a big Y, the big R could pair up with a small y randomly. These are the option, the possible combination that can take place. So once again, this going back to Mendelian genetics is to show you that the traits that you inherit is random, and during the development of the sperm and egg, it's distributed to each sperm and egg randomly. So here is Darwin's theory, his hypothesis. He said, for alleles, a pair of genes that carry the same characteristics are found in the same location of the chromosome. An organism has 2 genes from each parent. 2 genes, 1 from each parent. These genes may be of the same allele or different allele. A sperm and egg carries 1 allele for each trait because the allele pair segregate or separate during meiosis. When the 2 genes or pair are of different alleles and one is fully expressed, show up is another way of saying it, while the other is not noticeable, have not a noticeable effect on the organism, it appears that one would be called dominant. That's the one that shows up. The one that doesn't show up is referred to as being recessive, respectively. That's supposed to be quotation. So let's take a look. Let's see if we understand the theory, the gene theory. We have genes found on the chromosome. Let's see, how would you organize these? Take a second. I gave you on page 2 a section to write it on. Put them in order. Out of the 3, which one is the largest? Go all the way to the smallest. Okay? Tell me, which one's on top?
N is at the top, C is in the middle, and G is in the smallest.
So we have nucleus at the top, because inside of the nucleus you find the chromosomes,
In the middle.
right? And chromosomes are made up of your genes.
Yep.
What if I went smaller than that? What would the genes be made up?
The bases and then the atoms.
So that would be your DNA
Okay.
strand, DNA. Okay? All right, good job. So once again, your genes, during sexual reproduction, it creates variation. So all the slight differences you have, it's because of sexual reproduction. You have mom's information, dad's information, but you're not identical to mom and dad. You're a variation of them. And some of the things that led to that is crossing over. We saw that the last time. We see that parts of the chromosome actually break off and join other chromosome to give you a new sequence, a new combination. Gene linkage, there's certain traits that are always linked together. For example, if someone has freckles, most likely they have red hair, most of the time, right? If you have dark hair, what most likely will be the color of your eyes? Right? You have a darker color eye, brown eyes, most of the time, because those traits are usually inherited together, they're linked together. So here's a quick look, we saw it the last time, of crossing over. So here, let's say these are two homologous chromosomes, and so during that process of meiosis, pieces of the chromosomes actually exchange or break off, change location, and so you'll end up with a chromosome, homologous chromosome, that has a new set of information on it. And so that gives you a variation. And we see that again in gene linkage, two genes being linked together. And besides the color of your eyes and hair, you could see where gene linkage is important. Some genes will not get expressed unless another gene is turned on. So we are going to see that certain genes are relying on the activation of other genes. All right, let's move on to sex determination. We're talking about the development of that embryo. How did they inherit these traits? We're inherited through the genes in the egg cell, the sperm cell, the crossing over that took place in the production, fertilization. This is all creating or giving that baby all the different traits. Now, to determine will the baby be a boy or a girl is dependent also on the traits that you've inherited. So in sex determination, this is a karyotype or a picture of the traits being laid out. So in each section there, you get a chromosome from mom, you get one from dad, mom, dad, all the way throughout. So in a human, this is an example of a human, karyotype of a human organism. So we have 24 sets representing 46 chromosomes total. The last group, the 23rd pair, is what determines the sex of that child. Will that child be a male or a female? So if that baby inherits 2 X chromosomes, they will be a female. So that means they inherited an X from mom and an X from dad. Females can only give up an X chromosome. So as you could see, females only have XX. So mom can only, or egg cells will only have X chromosomes in it. Whereas a dad, for guys, let's take a look here, a boy, that 23rd chromosome will have an X and a Y. So a male can give up an X chromosome or a Y chromosome. So if that male gives up an X chromosome and it combines with a mom's chromosome, which is X, that creates a female baby. Whereas if the dad gives up, the sperm cell has a Y chromosome and it fertilizes the mom's egg, which has an X chromosome, that will create a boy baby. So now we're seeing how do we inherit male traits, how do we inherit being a female. So I have this little picture down here to show you. Here's the male sperm cell, 22 autosomes, but he's donating a Y chromosome for the sex chromosome. Here's the female egg, she can only give up an X chromosome. So once these two combine, that zygote now will have an X and a Y chromosome, meaning that this baby will be a boy, a male. Sometimes we can see disorders develop. We could see there could be a defect in the traits that's being developed. One of those genetic disorders is sickle cell anemia. So we see a lot of questions on this test. Sickle cell anemia is caused by an incomplete dominant allele. This is where a single nucleotide mutates, causing an abnormal hemoglobin in the red blood cell. So if this represents all the different types of blood cells, we reviewed the last session. So we have our white blood cells here. They have their irregular shapes, usual. Then we have the disc-like shapes representing our red blood cells. These guys here. The little ones here represents our platelets, and we went over the function of each last time. Here, look at some of these red blood cells. They are sickled shape. They're not the nice circular round shape. And that's because there was a mutation in the translation of the protein that makes up the hemoglobin of the blood. And we looked at translation the last time also. So because of that, we're going to see that this person will have difficulty in transporting oxygen through the red blood cell. And the way that mutation took place, if you take a look, we have the T, due to a mutagenic agent, change into an A. And so that's going to change the type of protein that would be made. In this case, the normal protein for hemoglobin has been changed. Another disorder is Down syndrome, which is referred to as a non-disjunction of chromosome 21 in the parent. So let's go back to this karyotype like I showed you before. And if you look at every group, we have 2 sets of chromosomes. But for a person with Down syndrome, they inherit a 3rd chromosome. All right, so this is non-disjunction, whereas in the separation during meiosis, instead of them separating equally, we see that they didn't, and one will end up with having more chromosomes in it than the other. And so there's also a link between this and older moms. We see as the age of the mom goes up, the chances or occurrence of having a baby with Down syndrome goes up. And so it's believed that the chromosomes don't separate as readily as they would in a younger mom. But with today's technology, we're able to diagnose and identify some of these disorders ahead of time. And with technology, we have moms that's having healthy babies due to the technology that we have available. Another malfunction I want to cover, because we see a lot of question on this, is cancer. Cancer referred to an uncontrolled division creating abnormal cell growth. And so during mitosis, we go from 1 to 2 cells to 4. In cancer, we see there's a rapid growth of the cells. And so they create a mass. And so what that does is disrupt the normal function of that tissue or of that system. Other diseases that you'll probably hear, a lot of times we see this in the read and comprehension section, is we have Tay-Sachs, which it's a defect of the lysosome. Cystic fibrosis refers to an imbalance in the result of thick mucus being in the lung and the pancreatic ducts. Many times we see these in reading passages in the test. Some of the sex-linked disorders that also show up in a lot of the reading passages, the reading comprehension section, is hemophilia, color blindness, and baldness. And so a lot of these are linked to the 23rd set of chromosome. So that's the sex link, the X and X chromosome. Here's some techniques that's used to diagnose if there's a disorder or not. And so one technique is referred to as amniocentesis. This is where they withdraw the fluid, amniotic fluid, while the mom is pregnant. Take that amniotic fluid that's surrounding the baby and examine the cells. Remember, the baby's still developing, so the fluid has a lot of cells floating around. So you're able to extract that fluid and examine the cells for any malfunction or disorders. Another technique we have been showing you here is karyotyping, which refer to an enlarged photo showing the pair of chromosomes. So, for example, you would be able to see Down syndrome because that's the extra chromosome on the 21st pair. So we'd use that for something, excuse me, like that. A pedigree chart, on the other hand, it's a diagram that traces the pattern of inheritance through generation. So when you're interpreting the pedigree chart, you have to remember the symbols. A normal female is usually circled, nothing shaded in. A carrier female has partially shaded area, normal males square, and the male that exhibits the trait is completely filled in. All right? Because most of those traits are carried on the X chromosome, the male, you'll see that the male will most likely will have the trait or not. Let's do a quick practice question on disorders. A child is born with a genetic disorder to parents who show no symptoms of disorder. Explain the type of information a genetic counselor might provide to the parents, and in your answer I want you to include these. Explain why the child would exhibit symptoms of a disorder, of the genetic disorder, even though the parent do not have that or show that disorder. How is it possible that the child would exhibit it? Identify one technique that can be used to detect the genetic disorder, and identify one type of genetic disorder. Let me give you guys again 2 minutes. Those who are joining us from home, you could use a Word document or on a piece of paper. Take a couple minutes to answer this. All right, so let's check our answers for that. The possibility, reasons why the child may express the traits even though the parents don't show it. It could have been where there was a mutation that might have taken place. The child could have inherited 2 recessive traits from the parents. There could have been some sort of non-disjunction where during chromosome separation, we see that one cell have more than the other. Next question, identify a technique. You could have named any of the technique we saw before. Amniocentesis, karyotyping, blood screening is possible. Using electrophoresis is another possibility. And any genetic disorders, we saw some today. Down syndrome, sickle cell anemia. You could have listed hemophilia, any of the other ones that we listed previously. Okay, so check your answers, see how you did on that. How are you guys doing? Any questions on any of those? Good? All right. All right, so that brings us into our biotechnology section. At the beginning, I said our technology have changed so much. We've advanced in technology. We're doing amazing things with genetics, and that falls under our category of biotechnology. A lot of the questions that you'll see on the state tests will focus on biotechnology. We are using it so much in our society right now that I'm going to say it's our hot topic for the test. Biotechnology is a method used to manipulate genes to develop new traits, combination, and a variety of organisms. So now we're looking at the fact now previously we just said that we're inheriting our traits from our parents, pretty much get what we get from our parents. Mutation may occur to cause some changes. We have crossing over, cause some changes. Now we're looking at where we can actually select traits that we desire, and we can manipulate them to produce traits that we want. And so ways that we could see that is artificial selection, inbreeding. So we see this in plants, where we could have plants that end up with traits that we actually want them to have, to be resistant to something or produce fruits the way we would like them to look or taste. inbreeding. We see this in animal, we see this in breeding of dogs and horses and so forth. Even though this was a natural process occurring in nature, but we're able to manipulate it in the way more than before. Genetic engineering, we're going to see where we can actually take one organism from the genetic information from one organism and insert it in the next. And cloning will also cover that, how we can produce an organism with the same trait that we're looking for. Right? Evidence or things that we need to focus on as a result of biotechnology, we can create plants that are resistant to certain diseases, resistant to temperature, dramatic temperature changes. We could clone things that we haven't cloned before because of biotechnology. We could create antibiotics. We can also use this to a way of diagnosing diseases. We could use it as a preventative treatment. We could use it as a control method. We could use it as a cure for plant and animal diseases. Let's see, artificial inbreeding. Let's take a look at each one. So this technique is used by plant and animal breeders to choose a desired trait for an organism. It's used to maintain, improve plants and animals based on the trait that that farmer or animal breeder would like. In inbreeding, this is where they mate closely related organisms to maintain that trait. Hybridization is where the breeders can cross two variation of species with that desired traits and hope that the offspring will contain that trait that they want. All right, the next group I want us to look at is genetic engineering. Genetic engineering is where we introduce genetic information from one organism into the next. And I know you guys have seen this one in class. So we talk about it when we were producing insulin. We have an animal cell will take the genetic information to produce insulin from the animal cell by using a restrictive enzyme. We'll cut out the code, the part of the DNA that we need that has that specific genetic information. Inside the bacteria has this plasmid DNA, and from that plasmid DNA, you could use the same procedure. By using a restrictive enzyme, you could cut out a piece or portion of the plasmid DNA. So it looks something similar to this. It's a ring of DNA. We can then insert the human DNA inside of the bacteria and then insert it back into the bacteria cell. It's easy to use a bacteria cell because they're simple organism, easy to manipulate. Not only that, they reproduce quickly and a lot. So we could get a lot of information from it. And so let's take a minute and review some of this in the video.
The plasmid DNA and the gene of interest are isolated from the host. An appropriate restriction site on the plasmid DNA is identified in order to insert the desired gene. The plasmid is cut at the restriction site and sticky ends are created using restriction endonucleases. The target gene is inserted into the plasmid DNA with the help of the enzyme ligase. This hybrid DNA is known as recombinant DNA. Recombinant DNA plasmids are introduced into bacterial cells. Once they are in the host cells, the plasmids are able to replicate. Eventually, the host cell divides. Some cells may have recombinant DNA and some may not. A test tube containing recombinant DNA in host cells may be grown on agar plates. The bacteria may be plated on a selective medium containing the antibiotic ampicillin. The host cells undergo cell division and form bacterial colonies. Transformed bacterial cells are resistant to ampicillin and show growth. Untransformed bacterial cells die because they are sensitive to ampicillin.
So we're going to see that there's some advantages and disadvantages to this technique. Genetic engineering advantages that we can improve nutritional qualities, where we could now insert maybe vitamins. If we want to reach a population that has vitamin deficiency, we can add nutrients into rice, for say. We could create resistant plants that are resistant to diseases or pests, or even making weed control. We can also improve survival rate of organism based on the environmental stress, like we could talk about cold temperature, severe changes. We could create plants that can live in that environment or be resistant to those environmental changes. New materials, we're able to produce new materials for factories and create new medicines. So the disadvantage of this technique is what we're seeing also is that insects are now becoming resistant to pesticides. And as they become resistant, they now could become a pest themselves, where we have to figure out a way, or can't find a way, how to get rid of them. We're altering the food, so we also see a rise in allergies because of the manipulation of the food, and also new diseases because of it. Any questions on genetic engineering? All right, so GMOs are animals that have been modified genetically, and we could see for those guys, their advantages is that we can use them to study diseases. We could create vaccines and test out vaccines safely in a transgenic organism. They could aid in the production of hormones and any pharmaceutical, in pharmaceuticals. We're also seeing where we can now increase milk production of lactase. We're going to take a quick break soon, in like another minute. All right, so just hang tight. And we also see increase where we can affect cholesterol. The disadvantage in that is that there's a production in certain medicines are rare and expensive. There's little scientific evidence of how effective it is. And there's also that ethical issue as far as gene therapy. Should we use it? Should we not? Who should use it? How it should be used? And also the effect that it could be a new form of terrorism. So just before we do a practice question, we're going to take a quick break. We'll return in 5 minutes. Okay? All right, let's take a break. From your break, we're going to pick up where we left off with a practice question. We just discussed genetic engineering and the gel electrophoresis process, where you isolate the gene that you're looking for. In this case, it's the gene for human hormone. You then insert it into a pre-cut bacterial DNA. So you could see here, remember all the cutting of the DNA is done by restrictive enzyme. You'll then insert it back in to the bacteria, and as the cell divides through mitosis, every cell after that will be identical to the original, but what's important is that it will have that human gene hormone in it. So I want you to take a second and answer this question. Name the process represented in this diagram. The original gene for the production of human hormone was most likely removed from A. State one possible reason why a gene from the production of a human hormone would be placed in a bacterial DNA. All right, so you have 3 slots for that. Take 2 minutes and write that down. Okay, so let's take a look. The process. What's the name of the process? It's being done? What do you guys have? Did you do any of the gene splicing? All right. So it should have been gel electrophoresis or genetic engineering.
Yeah.
Yep, I'm good. All right. The original gene for the production of human, that's a human hormone. So it's removed from the DNA of the human or of a human organism. Why would they put in a bacteria? Remember, bacteria, it's easy to manipulate, and they reproduce quickly and reproduce many. Okay, let's do another practice question. One variety of wheat is resistant to disease. Another variety contains more nutrients of benefit to human. Explain how a new variety of wheat with disease resistance and high nutrient value could be developed. So in your answer, based on that, first I want you to identify one technique that could be used to combine disease resistance and high nutrient value in a new variety of wheat. Second, describe how this technique would be carried out to produce a wheat plant with the desired characteristics. Thirdly, describe one specific difficulty other than stating that it does not always work. Determine one specific difficulty in developing a new variety using this technique. So once again, let me give you guys 2 minutes to work on that. All right, so 2 minutes is up. Let's take a look at the answers. For the first one, you could have used genetic engineering, selective breeding, cross-pollination. Any of those could have worked for the first question. The second one, describe how the technique would be carried out to produce the wheat plant with the desired characteristics. If you chose genetic engineering, then you could discuss that you are moving the gene from one organism to the other. If you chose selective breeding, you could say that you're mating two organisms for that desired trait. So understand the difference between the two, guys. Genetic engineering, you're actually removing the gene from one organism to the next, whereas with selective breeding, you actually have to mate, cross-pollinate the plant or the animal to get those traits. The problem with it in our last one is sometimes that process, the genes are not expressed the way that you would like them to express. It's not always an easy technique to pull off, and it's also sometimes difficult to isolate the gene that you're looking for. So double check your answers. See if you came up with any of those. Any questions? Okay. And so the last one on our list is cloning, and we're going to just quickly review cloning. So if we take a look at the images that we have here, I've pulled these images from past Regents exam, where they show here is the original organism that's being cloned. They would then take the egg cell, remove the nucleus from the egg cell, and then they would insert the body cell from the organism you want to clone. Once you've inserted in, then the cell, fertilized cell will then go through, remember cleavage, that whole process, it will go through that again. As it's going through that process, it will then be embedded into the organism that will carry the baby. And so the organism will carry the baby to full term. And so the baby's born. In cloning, you don't get an exact, visual replication of the original, because the clone has to start from a baby. What's identical is the genetic information, meaning that this offspring is genetically identical to the original, the organism that you are trying to clone. He is not identical to the carrier organism. He's identical to the organism you're trying to clone. And so we've seen cloning in other options. When we covered asexual reproduction, we could see in binary fission, we saw it, budding, same exact idea, where you have you end up with an offspring that's identical to the parent cell. So cloning is inserting DNA from one organism cell into an unfertilized egg cell from another organism whose DNA has been removed. All right, let's do some more practice question. Once again, I have you guys doing a lot of practice questions for this, because in the short answer, the free respond section of the test, there's a lot of short answers. So it's important that you get comfortable with this. So here we go. A scientist have successfully cloned sheep and cattle for several years. A farmer is considering the advantages and disadvantages of having a flock of sheep cloned from a single individual. Discuss the issue the farmer should take into account before making a decision. So in your answers, this is what you're going to answer. How a cloned flock could be different from an uncloned flock. What is one advantage of having a cloned flock? What is a disadvantage of having a cloned flock? Give one reason that the farmer could not mate these cloned sheep with each other to increase the size of his flock. What's a reason for that? That he wouldn't be able to mate the cloned sheep with others to increase the size of his flock. Last one, give one reason that the offspring resulting from breeding these sheep with an unrelated sheep would not all have the same information. So this is a little bit more detailed than the questions that we've been doing before. All right, so it's going to take a little bit more thinking. You're going to have to connect previous information to what we're learning now. All right, so I'm going to give you some time to think that through. This is at the bottom of page 5. All right, so let's take a look. At the bottom of page 5, you could fill in, check your answers. It says the first one, how a cloned flock would be different than an uncloned flock. They would all have the same genetic information. There'd be no variation in it. They would be identical to the original. There would be no diversity. An advantage to that would be the fact that whatever trait you were looking for, you would have it in this flock. A disadvantage to that is if they're all alike, what if there's a disease that affects one, it would affect all, because they're all exactly alike. One reason the farmer could not mate these clone sheep with the other sheep, most likely, not most likely, they will be the same, the one sex. Whatever sex you clone, that's what you're going to get in the cloning process. And a lot of them have a shorter lifespan, and their developmental difficulties in cloning. The last one is the reason, what's the best result for using a breeding technique? A breeding technique, you would have both parents involved, and you would have different gene combination, so you would get a variety of gene selections. Right, so you want to think about the advantages of cloning compared to the disadvantages in this question. So one of the sections I want to touch on before we leave, because we saw this in the Biodiversity lab, is DNA fingerprinting. DNA fingerprinting follows the process where you're going to cut the DNA using a restrictive enzyme. We use this for various things. You're trying to identify, look at different different organisms, and compare their DNA sequence. So oftentimes you'll see a well similar to this, where each individual DNA is placed. As they travel through the gel, they go from a negative to positive. I don't know if you guys remember, as they move through the gel, the smaller pieces move further and faster, and the larger pieces stay towards the top. You guys remember this from the lab, right? So you could use the DNA bands to compare them. So in a crime scene, you could see that the person that committed the crime in this sample would be individual C, because the bands are exactly the same or have the most in common. So if you look at this question here, this is one of the previous Regents questions I pulled up for you. It says, which male tiger is the father of the new cub? So you're comparing genetic information. So here's the father's information, male 1, male 2. Here's the cub. Which father has the most in common with the cub? Take a look, guys. Is it male 1 or male 2? Which one? Male one? Male one has the most in common, right. Okay, so that brings us to the end of our section of genetics. Remember, you inherit traits from parents, and you're inheriting the traits from the egg cell, the sperm cell. During that time, you get crossing over things to lead to variation. And so that's why you're a variation of your parents. There are genetic disorders that can occur where there could be non-disjunction or a base replacement. We saw that with sickle cell anemia or in Down syndrome. There are scientific techniques used to identify genetic disorders such as amniocentesis and karyotyping screening. And we're seeing that with biotechnology, we're able to manipulate genetic information to produce the traits in plants and animals that we want. Okay? Next, because of some of these techniques, we're seeing plants that are resistant, animals that are resistant to diseases or to certain chemicals. And so we're starting to see a new set of organisms develop in our environment. And that's going to take us into our section of evolution. So we see over time, things have changed. Over the years of the earth, we go from things being very simple to being more complex. We've seen in different sections. And we see that things are grouped based on what they have in common. And so classification is the grouping of organisms based on their commonality. So because a couple of the things that, if you do see questions on the test, we'll focus on the groups that they fall in. So you could be asked, what is the scientific name of a house cat? And so the way you would determine that is by the genus and the species. So the scientific name is Felis domestica. So from this, you could be asked a question, which is a larger group, the genus or the species? And you would see that the genus have more organism with those characteristics than the species. So evolution is the change of a species over time. You'll see biological changes and organic changes. And we're going to break it down into three categories. We're going to look at organic evolution, modern evolution, and the mechanism of evolution. The first area I want to start in is Darwin's theory of evolution, which is natural selection. The concept that organisms will have the traits to help them survive in a changing environment. Saying that if you have the trait that's going to help you to be best suited in that environment, you will be able to live and reproduce. And so Darwin said these are the principles you need in order to survive and reproduce. You need to be able to overproduce, make more than can survive. In case some die out, there'll be some species left to carry on that species. There's some organisms left to carry on that species. You're going to be the best at competition. It's called a struggle for survival. What are you able to do to help your species to survive? Variation is important. Within a species, there has to be different variation, different traits. So we see sexual reproduction give us variation. Even though we're all human in this room, we're all different. We have different variation, different gene combination. Natural selection, you need to be able to adapt to that environment. Survival of the fittest means that you were able to live and reproduce. Inheritance of traits, those helpful traits or those favorable traits is going to help you to survive. And it's important to also create new species, which is called speciation. So if we take a look at this schematic chart here, as we go through the process of evolution, it starts out like this. Formation of new traits within a species. That occurs through mutation, we just discussed in our last section. Have a new combination of genes. That creates variation within a species. That variation will then will give you the traits that's going to help you to be best at competing for food, live in space, and so forth. The organisms, some of the individual with the traits that are better suited for that environment, those ones will be then the individual in the population with that trait to survive. They will then multiply. Those without those traits will die off. And so that's our concept of natural selection. If you are better suited for that environment, you will survive and reproduce successfully. So that's the gist of the topic of evolution. As long as you could understand this idea, I want to say it's one of the easier topics, but you do get a lot of questions on it. Understanding each segment of this and understanding that you could get questions based on each section. Let's take a look at Darwinism.
Charles Darwin proposed a theory on how natural selection occurs. This is referred to as Darwinism, or the natural selection theory. Excessive reproduction, limited resources, variations among species, and survival of the fittest are the four guiding principles. Paramecia and oysters reproduce enormously, but there are not enough resources on Earth for all of them to survive. Hence, the struggle to exist arises. Variations occur both favorably and unfavorably within the animals. Examples of favorable variations include the aggressiveness of a dog and the ability of a deer to run very fast. Organisms that are more fit can survive and pass on their successful genes by reproducing, leaving unfit animals to perish. Sometimes, after the accumulation of many favorable variations, a new species may emerge.
So that idea of a new species emerging, it's not that this organism changed into this. A lot of times students believe mutation means one thing just changed into the other. That's not how it works. What happened is in this population, there were variation. The organisms in this population that had the trait to survive, live, and reproduce, those without the trait would die off. So those ones that live and reproduce maybe had some of the characteristics that this organism would have. So over time, we would see whatever that trait was to help them to be more successful in that environment. We would over time see that we would see more of that trait in the environment. And so as the environment changed, this organism here, those who had the traits to help them survive in the environment that's changing will live and reproduce. The ones that had more of this characteristics weren't able to survive in that environment over time. So what we see then is this organism. So we're thinking that this guy changed into this. That's not how it is. It's the fact that the genes in each group, whatever the genes were that was going to be best at helping you to live and survive, got passed on to the next generation to the point where we see new species in an environment. So you could come back over a period of time and you would see these guys instead of this. That's what it means. All right, so let's take a look at this. A hawk has a genetic trait that gives it better eyesight than other hawks at the same area. Explain how this could be led to an evolutionary change within the species of hawk over a long period of time. In your answer, I want you to explain competition within the hawk population, survival. Let's do them together this time. Competition within the hawk population. How would competition occur in this population? Who would be best at competing? Right. Better eyesight, hawks with better eyesight would be best at competing in this environment. Second, it says survival of various individual in the population. Which ones would be better at surviving again?
Would be the ones that have better eyesight.
The better eyesight would be best at competing. And so it says how the frequency of the better eyesight trait would be expected to change over time within the population. You would expect to see an increase in the population with the better eyesight. Then the next one, what would most likely happen to the hawks having the better eyesight trait if there was also an unusual weakness in their wing? I want you to think about this one for a second. Let me give you a few seconds. What would happen that even though they have better eyesight, if they had unusually weak muscle wing in their wings, what do you think would happen? So give a second, process that. Think about evolution. Right, so what you'll probably see in this section is that those with the weaker wings wouldn't have the same advantage. And they would then have to look for different ways to capture their food. Maybe we will see an increase in where they're preying on their animals from the ground, right? Instead of a higher level. They'll then have to live at a lower level. And so if that's an advantage for them, they will then live and reproduce. But if they're not able to take advantage of that, then over time you would see a decrease in their population and they would go extinct. Here's some recent evidence of Darwin's theory of evolution. Popular questions to see on the test is the peppered moth. You guys discussed this in class during the Industrial Revolution, where the barks of the tree were light color originally. So the light color moths were able to blend in. And then after the Industrial Revolution, the soot ended up in the air, causing the barks to get darker. So we saw the moths that were darker, they were now able to blend in, and the lighter color moths decreased because now they were able to get picked off and eaten, and so we saw a decrease in their population. So be aware that you will have to be able to explain questions that deal with environmental effect on evolution. Here's another popular one to see, the treatment of antibiotics. The fact that, here's day one, we have a bacteria that has the trait to be resistant to that antibiotics. And so we see the ones that are not over time die out, but the ones that are resistant will then start to increase because they reproduce quickly and the medicine is not killing them. So we could see over several days the resistant bacteria will live and reproduce. Over time we'll see in a population, same species, we would find a species of bacteria will then live and reproduce, and at this time they would all be resistant to the medicine. And this is why we have so many bacteria now that are resistant to antibiotics, and we're constantly making new antibiotics. Here's another concept of it. This is adaptive radiation, where we have geographic isolation. So remember, in the Beaks and Finches lab, we have it where the discussion is the animals from South America migrated to different islands. So you guys discussed the Galapagos Island, where that's where Darwin did a study and came up with the theory of natural selection. So as they ended up at different islands, due to maybe storm or drifting on wood for different reasons, the islands had different characteristics. And over time, these animals will develop into different species based on that, the characteristics of that island. So even if we were to take these birds from the island, or iguanas from each island, and bring them back to the original island, they wouldn't be able to interbreed with each other because they have developed into new species. So in that lab that you guys did in class, this is a picture of the adaptive process. The adaptive process. So the way you would read this chart is you would start from the middle, based on what food was available on the island. As you move out, you'll see the characteristics of birds that will live there. So a lot of questions in part D of the test on this wheel. Can you interpret this wheel? Okay. One of the popular questions you saw on that lab was you had to compare two. If the island had small ground finch and had, let's see, what was the other one? Small ground finch and let's take a small insectivorous tree finch, would they compete? And the answer should be no, because they have different food sources. So they wouldn't compete for food. So you have to look to see if are they competing for the same resources. That would increase competition. Right? So become familiar with this wheel. Be familiar with interpreting questions on this wheel. Right? Our next section of evolution is, how do we know that evolution is taking place? How do we know Darwin's theory? Do we just take some of those concepts? How do we know that things are changing over time? And some of the ways this takes us to our organic evolution, some ways through fossil evidence, that through fossils we can see things that used to be alive but are not today. We could tell changes that have occurred in the environment over time due to fossil record. There's comparative anatomy in the wing structure. So if you're following along in your packet, I've moved on to page 7. So these are some organic evidence. There's comparative anatomy. The fact that if you take a look at the horse, the cat, the bat, there's similar structures in them, leading or reminding you that they are deriving from a common ancestor, that as time changed, they evolved into different species based on them having that trait that's going to help them to survive in that changing environment. There are homologous structures where they both have wings, but they use them in different ways, and the structures are developed in different ways. Comparative embryology is the second one on that list on page 7. The fact that, take a look, we have a lizard, tortoise, pig, and human, but their embryological structures are similar, leading us to believe that they derive from a common ancestor. And as time went on, they developed into different species over time. There's similarities in the chemistry, the biochemistry of organisms, and it's called their molecular evidence. If you take a look at this tree here, we see the chimpanzee, the gorilla, orangutan, all derived from a primate, including us as human. We didn't come from them, but we have similar molecular structures, as in our ancestors were ancestral primate. But as times change and the evolution, the environment changed, different characteristics helped us to develop into different species. Here on this chart, we could see that there are different amino acids sequence, but we have a lot in common with each other, showing that we derived from a common ancestor. And so if you take a look at this chart, according to the diagram, is the pig more closely related? So where's the pig? Let's find it. Is the pig more closely related to the dog or to the kangaroo? Which one is he most closely related to? Is the pig most closely related to the dog or the kangaroo? Which one is it? Which one? Yeah.
It would be the dog.
And why? So it's related to the dog. And so here, it's right, so you're going to see that it's closer in the evolutionary process than the kangaroo is. Other reasons, besides them being closer on the tree, they have a recent, more common ancestor. So they're showing that they derived from the rabbit than the kangaroo did. So my clue, a good way to remember this, the closer you are on the tree branch with an organism, mean that you have a closer relationship with that organism. This is showing you two ways or two rate evolution is taking place. There's the punctuated or the gradualism theory, that sudden changes occurred over time or gradual changes occurred. More people believe in the gradual change over time. Right, and here's some other questions that you could see. Here it's showing, here's the common ancestor for all of them. Here's present time. So if organism J, K, and M is in the present time, what does that mean about these guys here below? They're dead. They're extinct. All right? Right. So maybe in fossil records and so forth. So you want to look at the chart showing the present time. And as we're seeing that things are evolving, one of the species that's been more successful in this process is the human population. And that brings us into our last section. Human population, due to the growth of industry, our advance in medicine, our population have grown past even our carrying capacity because we're able to maintain our resources through technology. We talk about technology, creating new things for ourselves, advancement in medicines. So we don't have that natural check that we saw like in the bubonic plague because of the advancement of medicine. And so with that, we're seeing that the human population is increasing without limits right now. And so because of that, we're affecting technology, we are affecting consumption, and that's going to disrupt or affect our ecosystem. So that takes us right into ecology, which is our last unit in this section. So in ecology, there's a couple sections in this, and actually before, because there's so much and I want to wrap it up, before we get into it, why don't we take another 5-minute break so we could just close out with ecology. So we'll break here and return in 5 minutes. Hi guys, welcome back from your break. We're going to finish up the last section on ecology based on the human population increasing. So we discussed that the human population is increasing due to less restriction, advances in industry, advances in medicine. We're going to surpass our carrying capacity. And so we're going to look at the impact it's having on our environment. I've condensed them into 3 charts. And so try your best to follow along. If you're watching this on video, you could pause and take time to go through each section. So we're going to look at how the human population is impacting water, air, and land. We'll start with water. The different ways that water is being impacted is that we're introducing toxic waste to the water, bodies of water, and these are different ways. DDT is an insecticide that was used in the past, and what would happen is it would run off from the plants into the stream, and it would end up into the water source. PCB is a toxic material that was used, and I think GE was a company that was reported the most as far as affecting the Hudson River with PCB. It was used in the generators, and what would happen, it would end up in the Hudson River, and PCB was a huge problem back in the days for the Hudson River. Toxic waste in general, untreated sewage, is another form of toxic waste ending up in the body of water. So whenever these chemicals end up in the water, they don't break down right away. It's difficult to break them down. They're not biodegradable. So what happens is they'll end up being absorbed into phytoplankton, small aquatic plant-like structures. And then once they're absorbed in it, then they'll be eaten. The plants are then eaten by tiny, like zooplanktons. Then that chemical gets absorbed into their body. And then those zooplanktons are then eaten by smaller fish. Then the chemicals that increase in the zooplankton will then increase into the smaller fish. The smaller fish by a larger fish. Each time the chemical is magnified as it's being passed through the food chain. And so what happens is the organisms at the top of that food chain has a buildup, a bioaccumulation of that toxic material in their body. And so the fish that's at the top of the food chain, or maybe it's a hawk or a pelican, they would then have the highest level of toxic material in them. So the negative effect of toxic waste ending up in the water is biomagnification. And we see the negative effect it's having on the food chain. At one point, the hawk or eagle population was decreasing in the United States because of this, because of the exposure to toxic material. Their eggshells weren't thick enough because of the exposure to the chemical. So every time they would sit on the eggs, they would break the eggs, and there was a decrease in the bald eagle population due to this. And so through conservation, they were able to develop, keep eagles and try to breed them and then release them back in the wild. We have then dealt with this problem by banning the use of DDT and PCB. So it's important that there are laws represented that will maintain the environment. So we have the Clean Water Act, and we'll just ban the use of some of these chemicals. DDT is used in different parts of the country because it's a good insecticide. So there's some countries that don't have a lot of money, they still use this to this day. DDT can also cause mutation because it's a mutagenic agent. Another type of pollution is thermal pollution. This is where factories will take water from maybe the river and cool off their machines and then redistribute it back out into that body of water. So the effect that it will have on the environment is the increase of temperature will decrease the amount of dissolved oxygen in the water. And because of that, we will see that aquatic life will be affected because there's not enough dissolved oxygen. One way that we can deal with this problem is through recycling that water. A third way we're negatively affecting water, and once again guys, these are some of the most popular types of questions that will show up on the exam. There are many different ways that water is being affected, but these are the most common questions asked. There's an increase in nitrogen and phosphate in bodies of water. That's called eutrophication, artificial eutrophication, and that's caused by fertilizers running off. So you're using fertilizers on the lawn, fertilizers to maintain crops that will run off into local bodies of water. And the problem that occurs from that is algae bloom. That's where you see that thick green fume on the top of the pond, and what happened is sunlight can penetrate through that buildup of algae. So all the plants underneath now will die out, reducing the release of gases because photosynthesis can't take place. The animals that rely on that gas will then die out. And I'll show you a video on that pretty soon. So one way we could deal with that is using less fertilizer. Oil spills are also a problem, and that's because of corrosion in oil pipes from the tankers. And so we hear about this from time to time, and how that affects the environment is that it creates death in these organisms, like large numbers. And the way we could do that is reduce our dependence on oil. So let's take a quick look at biomagnification, because it's one of the popular questions.
Due to industrialization and its related technologies, there is an increase in the amount of harmful chemicals in the environment. This includes chemical fertilizers and pesticides that are used for the improvement of crop growth and the dumping of heavy metal waste into bodies of water. Rainfall causes the fertilizers and pesticides to run off into rivers, streams, and ponds. This chemically polluted water is then consumed by humans and other animals and absorbed by aquatic plants. Once absorbed by plants, these chemicals can now enter the food chain, eventually causing toxic concentrations at the highest trophic levels. This phenomenon is called biological magnification.
Okay, so whoever's at the top of the food chain is affected the most by the accumulation. And we spoke about DDT. One way that we could deal with the reducing fertilization, it's not fertilization, wrong topic, fertilizer. We could use a biological control to get rid of pests, to treat any unwanted pests. I'm sorry, let me start over. Biological control is one way we could get rid of pests instead of using chemicals. And so an advantage to that is we won't have to worry about that chemical being ending up into our bodies of water. So using a pesticide, we wouldn't have to use that anymore by using a biological control. A biological control is when you use another living thing to control another living thing. So for example, like I said, ladybug and an aphid. You have aphids eating up a plant, you could introduce ladybugs, and ladybugs, they feed on the aphids, all right? Spiders. You could bring in different organisms to control the other. Advantages with the biological control, that ladybug is just going to eat the aphids. Whereas spraying the pesticide will then affect every organism, every plant in that area. There's less or no side effects, and it's cost effective. The disadvantage of that is that the organism you introduce may feed on other organisms that you want to keep. They also might be best at competing and outcompete other organisms. They may become a pest themselves because they're doing so good in this environment. They could live and reproduce in this environment. They may overpopulate and wipe out native species. they could bring diseases with them and could alter the ecosystem that you already have. So there are the pros and cons of using a natural control instead of pesticides. I'm going to skip over that. Let's go over to air pollution. Air pollution now. We see smog. All the ones on this list, guys, is due to the fact that we are burning fossil fuels. So when you're answering your questions on pollution, you want to look for any of the air pollution. Majority of them will be caused from burning fossil fuels. For example, take a look. Smog is due to cars and industries burning fossil fuels. That's your oils and your gases. All right, coal, same thing. Those are fossil fuels, and when they're burned, they release gases, greenhouse gases into the air. And so smog, because you're burning fossil fuel, we could see it affects the environment by there's an increase in respiratory diseases. So we see this more in our city areas, okay? There's an increase in respiratory diseases. What we can do about it is by using alternative fuels. We have different types of cars out there now. We have catalytic converters on the cars. You have to get your cars inspected annually to check the exhaust or the gases that's being released. Another negative effect of burning fossil fuels is when factories release sulfur oxide and nitrogen oxide into the atmosphere. What happens is those gases end up into the water cycle. So when it rains, those chemicals end up falling on the soil and in bodies of water. And because they will change the pH of the soil and the water, they will affect the organisms that rely on the soil and the water. So what we could do about it is reduce emissions from factories. We could have filtering systems, and this is just one way to get ahead of it. Global warming results also from fossil fuels being burnt. Fossil fuels end up trapping, increasing the heat in the atmosphere. And what we see of that, quick gist, we see melting of the ice caps. Not just because the polar bears are losing their habitat, but we're seeing an increase in flooding all around because the polar caps are melting. We see climate change is taking place. One of the things that's affected by changes in our climate is our food supply. The prices of food, we're not having things readily available to us as much because of it. And the main thing is that it's the prices are affected. Migration of animals into areas that you don't usually see them. Migration of diseases, malaria is showing up in areas that it wouldn't have shown up before, but because the temperature is higher, we're seeing malaria, for example, showing up in areas that we wouldn't normally see. So we're saying to move from burning fossil fuel to wind power, hydro, solar, geothermal energy, maybe using nuclear energy. All right, so just take a quick look.
Under normal atmospheric conditions, the temperature of the Earth is balanced by solar radiation striking the Earth, and then followed by the radiation of heat back into the atmosphere. Because of various types of human interference, an increase in CO2 concentration has occurred. This excess CO2 has formed a thick layer around the Earth's atmosphere, which has blocked the removal of heat radiation. This CO2 layer acts like the glass panel of a greenhouse, through which sunlight is allowed to enter, but whose excess heat is prevented from radiating out. This is called the greenhouse effect. Other gases, such as chlorofluorocarbons, methane, and nitrous oxide are also responsible for this greenhouse effect. This condition is causing an increase in the temperature on Earth, leading to global warming and the melting of polar ice caps and glaciers, which has led to a rise in sea levels.
And so the third one, I left it off the list because I want to focus on the cause for this being that chlorofluorocarbon, also known as CFC. It's ozone depletion. So this is the destruction of the ozone. So the ozone is there to filter out the radiation from the sun, and with it breaking down, what's happening is the sun rays are not getting filtered as it should, and so we see an increase in skin cancer and mutation. One way to deal with that is to ban the use of CFCs. Recently it said that we've seen some signs again of the ozone being depleted, but scientists are trying to figure out where or what's contributing to it. Let's go over to land pollution. So land, a couple things that I focus on. The most popular ones I've seen is overgrazing and deforestation. Overgrazing is where on farmland, where the farmers will have the animals just graze one area until it's completely, all the organisms, all the plants are removed. The problem with that is that it creates soil erosion, and it's difficult for those plants to regrow. And so a solution to that is to rotate the land usage, go from one area to the next. Deforestation is where we cut down trees for lumber, building, and etc. And the effect on that is enormous. We have it where deforestation can lead to a loss of biodiversity, different organisms in an environment, reducing or disruption in the food web. We could lose our opportunity of finding new medicine or new genes because we're cutting down trees. One way to do with that is you remove a tree to replace it with one through reforestation. Question?
Yeah, I kind of missed a spot on ecology section 1.
What page is that?
8.
Page 8? Which one do you want?
The line, fill in the blank.
Okay, so that's air pollution.
Okay.
So the question was in the packet, page 8, ecology, all of that, I'm sorry, that was water pollution. Water pollution. On page 9, number 2 is air pollution. And we're in section 3, which is, sorry, we did section 3, which was ozone depletion. And number 4 is land pollution. Good? Okay. All right, awesome. So reduction of biodiversity, that then takes us to the next page. The loss of biodiversity, the cause could be a natural disaster. Here are some causes. Natural disaster. The removal of a predator through hunting, for example, that can lead to loss of biodiversity. Human activities like cutting down a tree could lead to loss of biodiversity. Spreading of pesticides, that could destroy organism. Transport an organism from one area to the next. You could introduce a new species, and that can affect another species, the natural species, and that could create a loss of biodiversity. So introducing that new species is referred to a species that will come and take over because there's nothing to prevent it or stop it. It's called, those are referred to as invasive species. It's when a new species enters the ecosystem and disrupt or threaten the survival of the native ones. In doing so, the imported species may reduce that biodiversity. Okay, so let's take one of these quickly as a review. Let's take loss of biodiversity, since we just did that one. Those who are following along on the video, you should take some time. You guys have in the room have the paper list. You should take some time and go through each one and refer back to it and try to fill out all 4. So for example, let's do biodiversity. Biodiversity would be the answer for number 1. State how humans are creating this problem. You could say through hunting or over-hunting. Or you could have said cutting down trees. Let's go with that. Cutting down trees. Describe one specific effect that the problem you've selected would have on the ecosystem. It can affect our chances of finding new genes, new traits. It could disrupt the food chain. And state one specific action that we could take to reduce this problem. We could replant trees. We could put regulation on hunting, to transport invasive species, regulation at the borders, to inspect and regulate what's coming into our environment. Right, so those are some different ways. But I do encourage you to take the time to break down each one using the 4 questions here. And so we see that living and non-living things interact in the environment to maintain the ecosystem. And so the way they do that, here's the structure. Abiotic factors are all the non-living things in the environment. whereas the biotic factors are all the living factors. And so they rely on each other. So the living factors falls into categories. We have our autotrophs, which we mentioned before, the autotrophs. These are our producers. This is your plants. The heterotroph, these are the organisms that rely on other organisms to survive. The decomposers are essential to the process. They're the ones that keep the cycle going in the environment. In the consumers, them heterotrophs, could be broken down into categories. You have your primary consumers. Those are the herbivores. Your secondaries, these could be omni or carnivores. Tertiary, same thing, could be omni or carnivores. And so we just broke down each category so you could see them. So let's go to a food chain. So in this food chain, the primary source of energy comes from the sun. That energy is then transferred over to the grass, then to the mouse, to the snake, and to the hawk, to the eagle in this picture. Each stage, energy is lost because energy is used in order to consume. So if we were to create the energy pyramid to show how energy is transferred, using this food chain, what would be in the bottom? What would be the bottom of the pyramid? Okay, so it's energy, which one would have the most? And you'd also have more. So this is your, right? Your grass, and that's also your primary producer. Then who would go next as far as less energy? You have your mouse, good. Followed by, yep, and your hawk. Okay? So that shows each time. As energy is lost in the consumption process at each level, each trophic level. Any questions on that?
None.
None, perfect.
None. Just draw an arrow up.
Yep.
Draw an arrow up.
I didn't draw the arrow. Draw the arrow up. What goes on that arrow?
What?
What goes on that arrow?
I think the arrow would have,
What's decreasing as you go up?
energy.
Right. So as you move up, there's a decrease in energy. All right. And also you can use this to represent biomass. Biomass referred to as the amount of living things in that area. So you would find more grass in the area than you would find mouse. There would be more mouse than there is snake. So as you move up the pyramid, you can see the pyramid of energy and compare that to the pyramid of biomass. So we're seeing living and non-living things, but in this we just looked at the living things.
Also, body cells turn.
Exactly, more body cells as you move up. And so that explains why consumption energy is lost at each level. Let's take a look at this. Now we have a food web. It's a bunch of food chains put together. Let's see. Identify one population of organisms shown in the food web other than rabbit or mice likely be affected by the introduction of snowhawks, and explain why their population would be affected. So here we go. 1, identify one condition that might cause the snowhawk to leave the usual habitat of that area. What would cause a snowhawk? What would cause a species to leave their environment? What would make something leave? Right? Limited resources, food, mate, environmental changes. So in this example, let me see, level A and B, we already discussed, as you move up, energy is lost at each time. So in this, which one of these guys would represent your producer? What are your producers here? Level A are your producers. In level B, we have our primary consumers. So what would happen if, let's say I took the mouse out, who would be directly affected? Yep.
Snakes.
Right. Is that the only thing that the snake eats?
Yeah.
Yeah, so the arrow points to the consumer, points to the predator. So yes, the snake would be affected most directly because that's his only food source. And so there are different relationships that organisms have in the environment. There's mutualism, means that they both benefit from the relationship. There's commensalism, where one is benefited, one is unaffected. Parasitism, one is benefiting from the relationship, the other is not. And then there's that predator-prey relationship we just looked at in the food chain. And so materials are cycled throughout the environment, the non-living materials. So we see the carbon-oxygen cycle, where we see factories put carbon dioxide into the atmosphere. Carbon dioxide also leaves through respiration. And as it goes into the atmosphere, photosynthesis, plants use that carbon dioxide, and they release gases to help the organisms, and they release oxygen to help organisms. The organism will then release carbon dioxide and the cycle continues. So this is showing a connection between living and non-living. Same thing for nitrogen. We have nitrogen fixing bacteria that will actually change nitrogen into a usable form that can be used by plants. So that's where you get your plant protein from, because bacteria will take the non-living factors such as nitrogen and change it into a form that the plants could use. That whole process is referred to as the environment change over time, succession takes place. And let's take a quick look at it.
Due to changing environmental factors, the species in a particular community may be gradually replaced by others over time. This process continues, and successive communities are replaced in the same area, ultimately reaching to a final or climax community. The replacement of one community with a more mature one due to changing environmental factors and vegetation is called ecological succession. There are two general types of succession: primary succession and secondary succession. Primary succession takes place in an area that is colonized by organisms for the first time. This type of succession takes many years to reach a climax community. Secondary succession occurs in communities that have already undergone development but have been destroyed by a natural calamity such as a flood or forest fire. Since there's already fertile soil in this area from the previous community, it takes much less time for a climax community to emerge.
So I want to show you that as far as the environment change in itself, as an environment change over time, the plant succession also change over time, and this is referred to as succession, where you start out with your pioneer organism, like you have your moss, your lichens, and then that preps the soil for your shrub, your grasses and your shrub. They then prep the soil for larger trees, and the process continues until you see a forest. So there's aquatic succession, where a pond can change from a pond into a swamp because sediments will develop in the bottom over many, many years, and it can convert it into a swamp over time, which can then develop into a forest because of succession. And there's land succession, which you just saw in this video. So remember, guys, each session that we've done is just a quick summary on the key points of the state test. So you do need to know that you have to spend some time practicing the question. So I'll even suggest go back and look at session 1 as you're preparing for the test coming up in a few days. Go back, look at the key points. I just try to touch on the main topics that will be covered. If you have a review book given to you from your teacher, you need to then take each section and then go to your review book and do practice questions. At this point, my suggestion is to do as many practice questions. Those are the questions that I've pause throughout each session to review. You could also go online to Learn America, where you could see these videos on your own, and you could do some practice questions there also. And you could also view old Regents exams and do some practice questions based on that. So as you leave the last session, I want you to go back, reflect back on the topics we've covered, and try to do questions based on the main topics I've covered. So don't just take our review session as the only thing you'll have to do. You do need to spend some more time doing practice questions based on the main concepts. Okay, so thank you guys very much. Remember to prepare for the exam, go ready, bring yourself a dark pen. The Scantron will be filled out with pen, and you need to remember to sign the contract before you leave the exam so that it can be graded, and I wish you guys luck. Okay, thank you.
Expert teachers and educators at Learn America have created an online platform that helps students better prepare and improve performance in standardized tests in math and science. Step 1. Our unique methodology involves taking pre-assessment tests. Students take pre-assessment tests to determine their skills, knowledge, and readiness required for taking a course. Students are provided a detailed progress report at the end of the assessment to determine their strengths and weaknesses, and areas to focus on in order to achieve higher success in the course. Students access the courseware through the Learning Management System, or LMS, that is available anytime, anywhere, either through a browser or through the apps for the Android or iOS platforms. Students will have direct access to instructors, view any posted course announcements, and view a calendar of upcoming course events such as homework due dates, assessments, and so on. They may also download a learning report detailing any of their course activity, such as assessment scores, anytime. Step 2. Personalized learning plan developed based on the student's test scores in the pre-assessment test. The LMS includes an ebook for each course that provides theoretical concepts broken down by chapters and topics. Each chapter includes diagrams, graphs, or animated videos to provide visual demonstrations of the theory. The LMS platform has several features to make the learning process easy. Students can highlight important information, take notes, use the online calculator, and unit converter. Our expert teaching staff has created an extensive question bank that provides self-assessment questions in the form of quizzes after every chapter. These tests provide students a quick assessment of what they have learned in each topic or chapter. Students get to view the results immediately and provide an instant view of what specifically needs to be revisited prior to moving to the next topic. Step 3. Adaptive assessments and formative updates, where students then experience a series of assessments. Formative assessments are configured to reinforce the essential concepts behind major topic areas and to prepare students for more rigorous assessments as they march towards exam readiness. The assessment results allow students a full 360-degree view of the courseware to reinforce the learning, and step-by-step detailed solutions to reinforce problem-solving abilities. Faculty will use the formative assessment results to further guide the students. The students are then put through a more challenging adaptive assessment process. Much like progressing through a video game's levels, students climb the competency ladder by mastering one concept at a time. The adaptive assessments culminate in a mock final exam covering the complete course. Step 4. Interactive moderated online forum. Students get an opportunity to participate in online discussions. Here, they can post questions and share information with other students taking the course, as well as faculty and teaching assistants. Step 5. Progress report and intense interactive review dialogues. We provide a detailed, personalized progress report to view course progress. Students may participate in faculty-led review dialogues. This is conducted either in person or over an online conference. Step 6. Ready review. Towards the end of the course, as their test approaches, students take a final summative assessment that details specifically what sort of outcome they can expect from their exam. With this assessment, students can dig deeper into what they need to know. Practice exams are available anytime and can be taken any number of times.